Bright Tuty. Every student matters.
So the first part of the cell, which is plasma membrane or the cell membrane. It is the outermost covering of every cell. It is living, okay, so it is continuously growing. It is delicate, it is not tough like cell. Then it is elastic, it can change its shape. It is very thin and semi-permeable. This is important. Semi-permeable. Okay, permeability means if a wall, if there is some wall, some membrane which allows the movement of object, or all the objects, then it is called freely permeable. But in cell membrane, it allows the movement of only some objects inside the cell and only some can leave the cell. So that is why we say it is semi-permeable. It is a very important characteristic of plasma membrane, because only the desirable substances can enter the cell. Like a cell needs oxygen, so the cell membrane will allow the movement of oxygen. And the carbon dioxide, which is a waste product, it will allow the movement of carbon dioxide from inside to outside the cell. So this is the function of semi-permeability. This is because of semi-permeability of plasma membrane or the cell membrane. Structure, it is made up of phospholipids, that is the fat molecules, and proteins, these two things. Function. Firstly, it gives definite shape to the cell, okay, since it is the outermost covering, so it will give a shape to the cell. It acts as a barrier and protects the contents of the cell. All the contents of the cell inside, they have definite constant chemical composition. So if the cell wall plasma membrane will rupture, what will happen? It will lose its chemical composition and the cell will ultimately die. So it acts as a barrier and is for protection. Third, semi-permeability of the membrane allows only desirable substances to enter. The undesirable substances like bacteria, they cannot enter the cell. In some organisms like in amoeba, the plasma membrane is very flexible and it helps in engulfing the food. So the amoeba, the plasma membrane does not have any fixed shape. So if there is some food around, what will happen? The plasma membrane will form tiny projections around it and it will help in engulfing the food. So ultimately the food will just come inside. So this engulfing has been made possible only because of flexibility of this plasma membrane. So this movement as well as this nutrition is possible because of plasma membrane. Now, so next, it allows osmosis and diffusion of certain material. Osmosis happens when water molecules or the liquid content enter from outside to inside or from inside the cell towards the environment. And diffusion is movement of other particles like not necessarily liquid, it can be solid, it can be liquid, it can be gaseous, like oxygen, carbon dioxide, exchange of gases take place by diffusion process. Next, in some cells it is modified to perform different functions, like in small intestine it is modified for absorption. So all the cell membrane, the lining of the intestine, they absorb the nutrient. So these are some important functions of cell membrane. Next, let me take up the osmosis and diffusion across cell membrane in detail. Diffusion, I just told you, diffusion of gases, and osmosis in case of water. Okay, so suppose this is an amoeba cell, okay, and the oxygen concentration is higher outside and the cell needs oxygen for respiration. So what will happen? Oxygen will move inside from higher concentration to lower concentration. This is diffusion of gases. On the other hand, the carbon dioxide concentration is higher in the inside of the cell. So it will move from higher concentration to lower concentration, it will move outside. So this diffusion of gases helps in various processes because oxygen is required for respiration and carbon dioxide is a waste product which has to be excreted out. Then how do water moves? So it really depends. If water concentration is higher on the outside and in the environment, in the surrounding, and the cell needs water, then what will happen? Water molecules will move inside. This is called endoosmosis. When water is moving from higher water concentration to lower water concentration inside the cell, it is endoosmosis. On the other hand, if water molecule is moving outside, suppose if there is excess water inside, excess water has to be excreted out, then it is called exoosmosis. This was endoosmosis. Okay, so there are this osmosis, it leads to three situations, which is it's a very important topic from this chapter. So what will happen if a cell is kept in a hypotonic or hypertonic solution? Okay, so first is hypotonic. All right, so let's see. Hypotonic solution means a solution in which the salt concentration is less but the water concentration is high. Such a solution is called hypotonic. So if a cell is kept in hypotonic solution, the concentration of water inside is less. So what will happen? The water will move inside. Okay, this is called endoosmosis. I just told you. So the cell will swell up because it is absorbing more water, so there will be swelling up. On the other hand, next is hypertonic solution. In hypertonic, what happens? The salt concentration is high, so the water concentration in the surrounding is less. Such a solution is called hypertonic. So if a cell is kept in a hypertonic solution, inside the water concentration is high, outside it is low. So water will move from higher water concentration to lower water concentration. That is, water will move outside across the semi-permeable membrane. So what will happen? The cell will shrink. Since it is losing water, so the cell will shrink. So in hypotonic, the solution, the cells swell up. In hypertonic, the cells shrink. You have to remember this. Another case is the isotonic, in which the water concentration inside the cell and outside the cell is same. So whatever that, there won't be any net movement. Water will move outside, water will move inside, but the net movement will be zero. So there will be no change in the size of the cell. All right. Next, regarding this, there are two activities which will actually help you explain hypertonic and hypotonic solution. First activity is osmosis with an egg. First, wash 2 eggs with dilute hydrochloric acid to remove the outermost shell. There is a hard covering outside the cell. So once you wash it with dilute sulfuric acid or hydrochloric acid, it will be washed out. Now put first egg in pure water. Pure water contains higher water concentration and less salt concentration. So higher water concentration, that means it is a hypotonic. It is a hypotonic solution. So when you keep a egg in that hypotonic solution, you will notice the egg will swell up because of endoosmosis, because water concentration is higher in the surrounding but less in the inside the cell, inside that egg. So the water will move from higher concentration to lower concentration, from surrounding to inside the egg. So the egg will, you will notice the egg will swell up. On the other hand, keep the second egg in concentrated salt solution. This is a hypertonic solution. Remember, if salt concentration is high, that means the water concentration is less. If water concentration is less, then water will move outside. So it will lead to exoosmosis and cell will shrink. Okay. So in hypotonic, there was swelling. In hyper, it was shrinking. Another activity, take 4 potatoes and halved and scooped out to make a cup shape thing, and just name them, mark them A, B, C and D. Keep cup A empty. This is called control, because it will help us to compare different cups with the other potatoes with that. Next, in cup B, what you do, you put 1 teaspoon of sugar. So high concentration of sugar, that means it is hypertonic. Less water. Then in third cup C, you put 1 teaspoon of salt. Again, it is hypertonic. Either you put sugar or salt. In both the cases, concentration of water will be less outside. And in D, what you do, you take a boiled potato and put sugar or salt in it. So what will you observe after 2 hours? Obviously, there won't be any change in cup A because we did not keep anything. In cup B, what will we observe? we'll observe that in the scoop the water is filled up. Why? Because of exoosmosis. Since water concentration was less in the cup because we put salt, so the cells, from the cells in the boundary of the cup, the water will move outside and the cup will be filled up with water. In this also, the scoop or the potato cup will be filled up with water because of exoosmosis. But you will notice there is no change in cup D, because since we boiled it, so that cell was dead. Due to heat, the cell died. So the semi-permeability nature of the membrane was no longer existent. So it did not allow movement of water from inside or outside. That is why you will notice no change in potato D. Okay, I hope these two activities made it clear the case of hypertonic and hypotonic solution.